I'm Allison Jonjak, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual brown bag lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, I'm Tom Lochner, the Executive Director of the Wisconsin State Cranberry Growers Association. We're excited to be a partner with Extension to bring you research-backed resources, tools, and tips to keep Wisconsin marshes productive and profitable.
For the November Cranberry Virtual Brown Bag, we take a couple of months off for harvest, and then we group back together post-harvest to hear all through the growing season. We've heard a lot from our Wisconsin specialists, but we want to make sure that we're plugged into the rest of the nation and the rest of the cranberry growing world as well. So today we're going to hear from researchers out in British Columbia and in Massachusetts and in New Jersey to make sure that everything that is going on, we're getting to learn about and take advantage of and stay connected with. So we're looking forward. I want everyone to feel comfortable asking questions. As far as asking questions, you can do either the chat, which I'm monitoring and will ask on your behalf. You can also unmute yourself and ask a question directly. And if you want to ask a question anonymously, you can send a chat message that says just to Allison, and I will ask that question anonymously. So lots of ways to interact. I also want to mention that we need to remind there have been lots of tax changes in this year that will impact the next taxes you file. So I was just asked to say, if you have not yet talked to your tax specialist, you're a little bit behind the 8 ball. Please talk to your tax specialist. After those little housekeeping bits, I want to thank Tom of the Growers Association for helping host these. So Tom, if you want to say good morning.
Well, technically, I'll say good afternoon, and welcome to our continuing virtual brown bags. And thanks to Allison and Alex for putting these together. It's a great opportunity for growers to get together and take a little bit of time to learn on topics. So we try to hold these that have relevant topics, and at times that growers can join. I know a number of you are probably considering getting out into the woods or looking at it this weekend, and we wish you well with that. But I just want to say welcome, and we're going to continue to do these types of programs. We did have one thing to make folks aware of. We did start the process for developing the research program for the upcoming year with the research roundtable that was held on Wednesday, and we had very good participation, and I think we had a good session. That leads to the Wisconsin Cranberry Board annual meeting, which will be held, somebody correct me, but I believe it is the 29th of November. And but everybody should have gotten a notice on that, and there is a virtual option, so we welcome you to join or come down to the research station, join us at that. But just welcome everybody, and if you're going hunting, have a safe hunt, and hope you and your family have a great Thanksgiving.
Thank you. Thanks to feedback from the education committee, from growers, and from researchers at Wisconsin, I kind of took a little straw poll of who we really wanted to hear from at these brown bags. And a reminder that if you ever have someone you want to hear from, let me know, and I'll do my best to book them in. But we had questions about pollination deficit, which is an area of expertise for Sandra Gillespie at the University of Fraser Valley. We wanted to hear Jeff Neyhart is new at the genetics program in New Jersey, and so we wanted to hear what he's looking forward toward doing. And then Giverson Mapombi has been doing a lot of work in fruit color and quality. And fruit color and quality also came up a lot at our research roundtable on Wednesday. So those are important and interesting topics. So I'm hoping everyone is looking forward to hearing them. We're going to go through in that order.
Good. Well, hello, everyone. Thank you for having me here today. I'm going to talk a little bit about my new program with USDA-ARS, or Agricultural Research Service here in New Jersey, focused on cranberry breeding and genetics. So I'm very new in this position. My program started only in February of this past year. So I'm not only new to this role, but also very new to the cranberry community. My background is in small grains, actually. But I'm hoping to apply that knowledge to in this role of cranberry breeding and genetics. This presentation will be light on results, but I'm trying to be very high level about what I intend to focus on. And so just to talk about the objectives of my program, really it comes down to breeding improved cranberry lines to support variety development. And this is also known as pre-breeding or population improvement. Really, it's that upper stream stage that supports the eventual variety development programs at Rutgers and at Wisconsin, and then also in collaboration with folks at Massachusetts and in programs beyond those 3 states too. My focus is on improving fruit yield and quality, improving resistance to diseases and other pests, and then also tolerance to heat and other abiotic stresses. And I'll talk about how I'm really diving into 3 big projects, these big 3 Ps of my program that I'm using to help address these objectives. One is in more predictive breeding approaches and technologies, another in high throughput phenotyping, and then all in the context of pre-breeding and genetics research. So I'm just going to give a high level overview of these 3 projects, the first being predictive breeding. Cranberry being a long-lived woody perennial lends itself to a very long breeding timeline where there are not only biological constraints, but also some resource limitations. First, the process of going from making a cross to generating new genetic variation to propagating out seedlings or plants to establishing those plants in a bog or marsh setting and phenotyping those plants over multiple years. And then finally to variety release could take anywhere from 10 to 30 years, which is an extremely long time considering some of the challenges we're facing in terms of new and changing environments, new and emerging pests, and trying to maintain fruit yield and quality, where we'd really like to shrink that timeline down so we can start to evaluate new genetic variation and hopefully select more intensely and precisely to develop new varieties that will meet grower needs and be improvements upon existing varieties. And so there's biological constraints from going from seed to seed, and that's really the driving force of breeding, and going from one cross to another. It takes a long time to establish experimental plots. And then just as it is a large investment to, say, renovate a bog or a marsh, it's a long-term investment to establish field trials over multiple seasons. And it's important when making the decisions of what goes into a bog or marsh that we're choosing the right material that is going to have the best potential for a new variety. And so my program is examining new approaches that hope to make the decisions about what plants to evaluate in the field setting a bit potentially more precise and reduce that time. And so we're exploring some genomics-enabled selection methods where we can use DNA markers throughout the genome on new variety candidates to actually make predictions of how they're going to perform before we even evaluate any of them at the bog or marsh scale. And using those predictions, we can make selections upon those new those potential varieties and plants, and meaning this is a potential resource saving for breeding programs, where instead of allocating a huge acreage to many new genetically distinct plants, we may use these marker technologies, this DNA sequencing technology, to make selections and cut that resource allocation in half. So now we're allocating more time to fewer, more promising variety candidates, and that means we can focus on them more intensely and evaluate their yield and quality potential earlier on. It also means that we can go from one cross to another in a shorter amount of time. Instead of waiting those 10 to 15 years before we know a variety is promising and we can use it as another parent in another cross, we can use these predictions based on genomic information to make those same selections. So instead of that 10 to 15 years from across to across, maybe we're thinking closer to 6 years or 5 years. And that means more rapid generation of new genetic variation that we then select upon for the next cycle. All this is in an effort to reduce that breeding timeline and get to those more promising new varieties sooner. The second thrust of my program is in high throughput phenotyping. So we know in breeding, many traits that are really important are time consuming, laborious to measure, or are measured subjectively. For instance, we measure yield by harvesting berries by hand. We use visual ratings to assess fruit rot severity. We're using time-consuming and expensive ways to measure fruit quality via chemical analysis. And then also there's a resource constraint to all of this, because due to person power or time, the number of observable and harvestable breeding plots is limited. And so we need to, one, make sure we're taking advantage, or we're assessing the most promising varieties and allocating those resources more intensely. But then also we want to expand the scope of how many plots that we're evaluating in a given year and how much data we're collecting on those plots. All of this is to make more informed selections. So my program is evaluating sort of high throughput methods of generating phenotypic data. And one approach we're taking is developing this phenotyping cart, which will traverse through the breeding plots here in New Jersey, and then hopefully this technology can be applied elsewhere to gather phenotype data at a much larger scale. And in one way we're doing this is through image-based phenotyping, where we've mounted RGB cameras to this phenotyping cart. And as it traverses through the breeding plots, we're taking overhead images of those breeding plots, those cranberry beds at different time points during the growing season. So for instance, we're using this information and new sophisticated computer modeling and machine learning techniques to try to identify in these images where are the berries, how big are they, at what level of ripening are they, what is the, essentially get these proxies for fruit yield and quality using just images. And this technology has advanced so much in the last 10 years, these computer vision based techniques and machine learning based techniques, that we can do a pretty good job of trying to identify and separate out and gather new information from simple images like this. So for instance, on the left-hand side, we have an example image from this phenotyping cart, and you can see there are berries scattered throughout. And on the right-hand side is essentially just one layer of that image. It's just a mask of the pixels that are occupied by berries. And from this sort of information, we can get to making estimates of how many berries are in this image, and potentially get to information about their size and stage of ripening. All important pieces of information. So what this also means, though, is that we can, instead of the more manual processes of making ratings like this by eye, we can use computer vision and image-based phenotyping techniques to phenotype many more plots than we might possibly be able to cover as a single person. All of this means we're getting more information on more diverse cranberry potential varieties, and so we can make more informed selections about which are superior for our growing region and then elsewhere. And so we're not just looking at potential yield and fruit quality based on these images, but also potentially using this as a proxy for rating fruit rot severity, looking at uniformity of flowering and fruit ripening, and then using other sorts of image-based technologies to look at things like heat stress incidence and potential tolerance to those abiotic stresses.
We've got just a couple minutes before we head over to Giverson. So if you've got key points you want to hit before the end.
Oh, sure. Yeah, I can. Just the last thing is all of this is in the context of pre-breeding, which is this upstream stage of plant breeding where we're trying to look at cranberry wild relatives as a source of useful variation to improve things like adaptation to heat and cold stress, potential new fruit quality traits, resistance to diseases or pests. But some of the questions or one question that my program is examining is how do we really identify and exploit that source of new genetic variation for improving breeding programs. And so I've actually, I'll skip ahead here. So really just remember these 3 takeaways about my program. 1, we're looking at predictive breeding to accelerate improvement, phenotyping to increase the scope and scale of understanding complex traits, and then pre-breeding to harvest that novel genetic variation to support new variety development. So if there's any questions, I'd be happy to answer them now, or we can kind of wait until after Giverson's discussion.
I haven't seen any come into the chat yet, but one question that I have is, I'm sure the Wisconsin growers will love to hear how you plan to work in support of Juan and in conjunction with other researchers that we've already been working with, and how your project will kind of fit into the whole ecosystem of cranberry breeding.
Yeah, so like I mentioned, this is more upstream breeding, I guess. Juan is working on variety development. Nick Borsa here is working on variety development, and my program is intended to support both of theirs through developing these improved. It's more populations of cranberry, not necessarily varieties. They just may have unique traits that could be integrated into Juan's program or Nick's program to develop the next generation of varieties. And so my research will be really deeply involved with others working on plant pathology and physiology and entomology to really that taking a step back into new genetic variation to try to identify new traits that could be useful for both variety development programs.
This seems to me to be really exciting in that it's going to help everybody cut years off of their programs and come to better results in faster time. So I think it's really, really great to have you in your role here.
Yeah, that's my hope. And just because I'm based in New Jersey doesn't mean I'm not helping to support growers there in Wisconsin and in Massachusetts and nationwide.
Amaya asks, where will you find the genetic diversity needed to achieve the pre-breeding focus? Genetic variation is so low in many of our current cultivars.
Yeah, part of that is looking at current germplasm collections. So for instance, Rutgers maintains a pretty diverse germplasm collection, but this also means potential new collections of wild cranberry from its native range, and then also looking to the secondary gene pool of related species that are still crossable and can be intermated with cultivated cranberry to bring in some of that useful genetic variation.
Awesome. And another question that we had heard about on Wednesday at our research roundtable was, how does this fit in with Juan's trait mapping research across regions?
Well, I guess, so I'm working closely with Juan to try to maybe use our location as another New Jersey, that is, as another site where we can test material coming out of his program. And I'm really interested in understanding how environment and genes interact to produce the phenotypes that we see, and how we can use that information to make more informed selections about targeting potentially locally adapted varieties to different growing regions and different environmental conditions.
Awesome.
So I hope that answered it.
Yeah. And we'll have, again, more time for questions for all 3 of our speakers at the last 10 minutes of the hour. But thank you so much, Jeff, for this overview. And I know you've just started in this year and you've gotten a lot done. And I'm looking forward to seeing, everything that comes out of your research. So glad to have you.
Yeah, thanks.
Giverson, if you want to share your screen.
Good.
Fantastic. A little bit on color and quality.
All right. Good afternoon, everyone. Thank you for the opportunity to come and present some of the research that I've been doing for the past 2 years. Since I'm new to many of you, I'm going to start by giving a brief overview of some of the research projects that I've been doing. I started my position in 2019, and I do applied research focused on plant physiology. For those of you not familiar with the Zimbabwean accent, I try to put as much information on the slides as possible, so if you can't follow me. All right. So one of the things that I'm actually working on is the evaluation of new varieties. Here in Massachusetts, we do not have a breeding program like you guys in Wisconsin or in New Jersey. So I've been working on the evaluation of new varieties and trying to give some of our growers performance data that is based on our conditions. Another project that I'm currently working on is agrivoltaics. So agrivoltaics is the co-location of agriculture and renewable energy production. We started this project in 2019. We had those plywood solar panels that you can see on the left. And currently we have a grant from the United States Department of Energy, where we're actually installing a bigger project where we're looking at agrivoltaics in Cranberry, but we're also looking at other projects in vegetables as well. So that's one of the things that I'm working on. The third thing that I'm really interested in is the investigation of canopy management as a cultural practice to improve fruit color. So I've been looking at mowing, pruning, and sanding just to see how they improve light penetration into the canopy, as well as how that improves the fruit quality as well. So I'm going to talk a bit about that at the end of the presentation. One of the main things that I've been looking at in my program has been the evaluation of plant growth regulators. So with my research, I'm trying to use plant growth regulators for a variety of tasks in cranberry production. Because I come from a tree fruit background where they use plant growth regulators for everything from fruit set to quality. That's what I'm trying to replicate the same in cranberry. So I've been looking at improving fruit quality. I've looked at color and firmness. Currently, I've discontinued the work on fruit firmness. I've looked at improving fruit set in cranberry. I've also looked at controlling excess canopy growth. And then I've also looked at improving the canopy establishment after the bog renovation. So those are some of the projects. Most of these are still like early projects, just exploring the use of plant growth regulators. So I'm now going to talk about the improvement in fruit quality, focusing on some of the color research that I've been doing. So like I mentioned before, I started this research in 2019, and my objective was to see if I could improve fruit color using abscisic acid. So what I did was looked at different application rates and timing of abscisic acid to see if that can be used to induce andocyanin biosynthesis for optimum red color. The idea for the research came from previous work that has shown that the application of ABA at verizon, which is color break, has been shown to improve red color in grapes. So I thought maybe we could get similar results in cranberry. The picture on your right shows that the differences in color between treated and non-treated grapes. As you can see that abscisic acid actually works to improve food color in grapes. So my question was, would it work for us in cranberry? So in 2019, the research that I did was focused on just 2 cultivars. I did Stevens and House. Then I looked at 3 concentrations, which and also did 2 applications, which was a single application at color break or a double application at color break and halfway between color break and harvest as well. So these are the results from 2019 now on fruit color in Stevens. This graph shows the total andocyanin content and then shows the difference between control and treatments. As you can see, there were no significant difference, which was a bit disappointing. So I was expecting this to work in cranberry. And then I also looked at the results in house. I also looked at the results in house, which were very similar to what we observed in Stevens. There was also no significant difference in total anthocyanin content between the treatments and the control. So my question was now asking myself, like, why didn't ABA work in Stevens and House Cranberry? So going back into literature, I looked at some studies that were done in the 1980s and early 90s. It shows that the surface morphology of cranberry cuticle is not very conducive for penetration of sprayed chemicals. So apparently cranberry has a very thick cuticle that doesn't allow rapid diffusion. So based on this, my conclusion was now that the future studies should focus on finding ways of improving penetration into the cuticle. And then I was also going to try other plant growth regulators. That was based on the initial study that I did in 2019. So moving on to 2020, what I did was to increase the number of plant growth regulators that I tried. So I added ethifone and jasmonic acid. And with ethifone, I ended up with two different formulations. So I'm just going to call them ethifone A and ethifone B. And this was supposed to have different uptake effects, so they were different in their formulations. The ethifone was applied at 4 and 8 pints per acre. Then the jasmonic acid at 280 and 480 parts per million. The abscisic acid at 1,000 parts per million. These were applied with the recommended adjuvant, which was just a wetting agent for the ethifone, as well as a non-surfactant, non-ionic surfactant for the jasmonic acid and abscisic acid. These experiments were then repeated with 3 other adjuvants that I'm going to talk about eventually. So last year, because we didn't have a lot of people helping us because of the pandemic, I only did this experiment on Stevens Cranberry. So in terms of expanding the adjuvants, because one of the things that I wanted to try to do was to try and increase the penetration. After some consultation with people in the industry, I came up with 3 additional adjuvants that I wanted to test out with my plant growth regulators. So these are Herbimax. This is a premium oil surfactant that has been shown to increase herbicide activity. I also looked at MSO, and this is a methylated seed oil that has been shown to increase penetration without disrupting the cuticle. And then also at Veda, I looked at Veda, which is a proprietary adjuvant that has also been shown to increase penetration. So the idea between trying these different adjuvants was to try to see if I could get anything that could improve the efficacy of the plant growth regulators that I tried. And then another thing that I did in 2020 was instead of just the traditional TACI analysis, I also did the DGI analysis with ocean spray. So this is an image analysis where you look at fruit color, then it gives you 5 different classes. For those not familiar with the DGI output, this is the image that you get from ocean spray. And this is an image, this shows 5 different classes. You've got class 5, which is a deep dark cranberry pixel. And then you have a white or green uncolored pixel. So that's just the image that you get from the DGI analysis. Moving on to the results from 2020, these results were actually very encouraging compared to what I saw in 2019. So the application of ethifone, all the plant growth regulators that I applied except for abscisic acid in conjunction with Herbimax, they were very efficacious in improving total anthocyanin control. And in the best treatment that we have, which is the ethifone at 8 pints per acre, this was actually like a 71% increase in total anthocyanin content. And you could also see there was a red effect where in terms of the highest concentration of ethifone at 8 parts per acre was very efficacious compared to the 4 parts per acre. So this was very encouraging to see. And then moving on to the data from the DGI. So one thing I should note is that according to Ocean Spray, there's no correlation between DGI and TACI. What I did with the data from DGI, this graph that I have, it shows the percentage of fruit in each color. And if you look at the top, I also looked at the percentage of fruit above class 3 because I assumed anything above class 3 is what is considered desirable for us. So for example, in the control, we only had 94% fruit above class 3. But if you look at one of our best performing treatments, which is the Ethaphone 8 pints per acre, you had 98% fruit above class 3. So that's a 3% increase in fruit above class 3. And those, all our top 3 treatments were the ones that had Ethaphone in them as well. So this is just to give you like an actual image that you get from DGI. So on the left is the image from the control, and on the right is an image from the Ethaphone at 8 pints per acre. You can actually clearly see the difference in fruit color from the treatment. So you see how it works by getting a better color from that. So looking at the other experiment from this is with the MSO, or the methylated seed oil. With this, in this case, only the Ethaphone treatments significantly increased TACI compared to the control. The abscisic acid and the jasmonic acid was not very efficacious. And you could also see the red effect, where the higher rates at the 8 pints per acre, they had better results compared to the control. So that was really good to see as well. And then in terms of the MSO, if you look at the DGI results, in this 88% fruit above class 3. And if you look at our best performing treatment, that was the Ethaphone 4 pints per acre, that had 95.8% of the fruit above class 3. So that was really almost a 7.1% increase in fruit above class 3. So that was really encouraging to see. The combination of MSO with the adjuvants that we tried was also very efficacious. I'm not going to show the results from VEDA and the other adjuvants because of limited time, but based on all, after looking at all the results that I got, so what I found that what works best is the Ethaphone B, the formula, because that's a different formulation, and then the Ethaphone A, I mean, it's almost similar. Then you also get the abscisic acid and the jasmonic acid as well are working. In terms of the adjuvants that I've tried so far, I think Hebimax is proven to be the best, and then you have MSO and VEDA, and then also LI-700. I didn't include it here, but LI-700 has also been shown, I mean, it's also been shown to work very well in my experiments as well. So building on that, what have I done now in 2021? I do not have time to show those results, but what I've done is I've reduced the number of treatments from those that proved most efficacious in 2020. And what I've also done is I've now increased the number of varieties that I'm testing. So I have Malika Queen, I have House, and Stevens as well. Unfortunately, this year for us, I don't know, in Wisconsin, we have high fruit rot incidence. So I think that's going to have a big effect on my results. The image on the picture shows one of the samples that I picked. As you can see, there's a quite high incidence of fruit rot, and I think that's going to affect the results that I see this year. Also this year for me, I'm trying to figure out the chemigation. For us in Massachusetts, here, we still use a lot of chemigation. So I'm trying to figure out if we can apply these plant growth regulators through chemigation. So what can we do now? Because this research with plant growth regulators is still going to take maybe 2 more years just to figure out the treatments and maybe hopefully get some registrations. Based on some of the work that I did with the canopy management, what I found out is that although not significant, pruning your canopy to just open up the canopy and let the light through actually increases TACI, or increases the total anthocyanin content. I also did a similar analysis with Gina. So this is things in my collaboration that I'm doing with One, where we were just looking at the image analysis. This was before DGI, and we've got similar results where if you do canopy manipulation, you can actually improve the color. In this case, the mowing treatment was not like the very severe mowing that we have. We still left some fruit on the canopy as well. So canopy manipulation could be one of the things that you could do right now if you have a thick overgrown canopy, just to try and improve the fruit color as well. And then this is some data that I got from Thierry at Rutgers in New Jersey. He showed that there's a negative relationship between increasing weed biomass and fruit color. So this graph shows fruit color ranked from 0 to 10, of which 10 is a deep red color. as your weed biomass increases, you get a reduction in your fruit color. So this makes sense because especially if you have the kind of weeds that shade the canopy, you could actually be shading because you need light for the formation of anthocyanin. So weed management could be one of those things that you currently do right now, especially if you have those weeds that have the propensity to shade your canopy. So that's one of the things that you should be able to do now. Like I said, I've done some other work with feminists. I didn't get great results with that, so I don't think there's any need to share with that, but I think this is all that I've been doing so far. And I'm right now I'm expanding this work to look at more ethylene precursors as treatments for plant growth regulators as well. I would like to thank all those people that have helped me in my lab. There's been a lot of people that have helped me, as well as some collaborators from Wisconsin, one in Matthew, and as well as Ed, who has provided some of the varieties that we are testing in Massachusetts as well. So that's really exciting for us, and thanks to him. And then all the funding that I've been getting from the cranberry industry, as well as the collaborations from Ocean Spray and some products from the following chemical companies. Thank you very much.
Thank you. I will take a couple of questions specifically for Giverson, since Sandra and Jeff both had a couple of individual questions, but also now's the time to start asking questions from anything that you've heard during the presentation, including questions of your fellow growers. I appreciate everyone sharing. I guess I can put my screen back on for the final few minutes here. Yeah, we're in peak question hours, so I would love to hear. We had, I guess, a few. All right, so Giverson, we are coming up on the end of the hour, so I'm going to make one more reminder. If you haven't spoken with your tax preparers yet for the new tax changes coming for the coming year, please do that so that you are ahead of the game. So thank you, Tom and Alex, obviously the University of Wisconsin Division of Extension, but then a special thanks to Sandra from Fraser Valley University, to Jeff from the USDA in New Jersey, and then from Giverson from UMass. Thank you guys all for connecting with us today and keeping us abreast of everything new in pollination, in our pre-breeding genetics program, and in fruit color and quality that's going on throughout the world. Great thanks to everyone.